Hi, my name is Chelsea Hayes, and this is our presentation on Alzheimer's disease and cerebral amyloid angiopathy in the Octodon Degu. Alzheimer's disease, AD, is a neurodegenerative disease characterized by amyloid beta plaques and neurofibrillary tangles that lead to cognitive decline. Additionally, several studies have identified an interplay between cerebrovascular diseases and AD. Most prominent among these is cerebral amyloid angiopathy, CAA, a condition present in over 90% of AD patients, where amyloid beta plaques are deposited in vascular structures of the brain. Associated with CAA are astrocytes, a glial cell involved in blood-brain barrier regulation. We propose astrocytes, blood vessels, and amyloid beta plaques as markers to investigate CAA. The predominant animal model in current AD studies are mice, which do not naturally manifest AD-like neuropathology. Since a large portion of transgenic mice artificially manifest familial AD, which accounts for less than 5% of human AD cases, the AD research field has encouraged the investigation of novel animals that naturally model the remaining 95% of cases, known as sporadic AD.
We use Degus as our animal model instead of AD transgenic mice. Octodon Degus are a diurnal, social, and long-lived rodent endemic to Chile, which offer a promising alternative to mouse models for AD research. Unlike mice, the Degu naturally develop AD-like neuropathological features such as A-beta plaques and neurofibrillary tangles, and in previous studies have been seen to exhibit a CAA-like phenotype. The spontaneous manifestation of AD-like pathology coupled with their longevity makes the Degu an attractive model for longitudinal studies and better modeling of SAD observed in humans. We employed the Life Canvas protocol for this project. The platform uses a rotating electric field that accelerates tissue clearing via delipidation and allows for immunolabeling of CAA markers in deep tissue regions, a technique that has never been attempted previously for Degus. The transparency of the tissue then allowed us to capture 3D cellular resolution images of the brains via a light sheet microscope.
Now for the results. Figure 1 is a 3D render of Degu brain after Life Canvas protocol. Figure 2 shows LEL, GFAP, and 6010 staining for blood vessels, astrocytes, and A-beta plaques respectively, for both the older and younger models. Prominent differences are seen between the different aged models. Figure 4 shows the amount of A-beta plaques in vessels plotted for both age groups. We see that the old group has more vessels containing A-beta as well as larger A-beta plaques within those vessels, based on linear mixed effect statistics, showing that the amount of A-beta containing vessels increases with age. Figure 5 shows the size of plaques in vessels based on brain regions. Comparing young versus old, we see that the thalamus has significantly more vessels containing larger A-beta plaques in older animals compared to the younger ones, indicating that specific brain regions in Degu are more prominently affected by CAA with age. These results show that the Degu are not only a valuable model for AD, but also for CAA. Thank you for listening. Here's a QR code for more information.